Hello and welcome back to this scientific ethics lecture where we're going to continue again with our stem cell policy. So in the previous lecture, we take a look at kind of the insurance coverage, who's paying for kind of experimental treatments, especially as it relates to stem cells. Now what we're going to look at are some of the rules associated with stem cells. And so we're going to get into some of the kind of ethical dilemmas that are associated with stem cell policies. Now, to start out with, the kind of most prominent policy or law that's in place related to stem cell science is the Dickey-Wicker Amendment. And this is an amendment that was originally signed by President Clinton that essentially bans federal funding for the creation or the destruction of human embryos for research. And so this is basically something that goes into the budget. It's described as an appropriations rider, but it's basically kind of an amendment to the budget for the US Department of Health and Human Services, which includes the National Institutes of Health, which provides the primary source for biomedical research funds within the United States. It basically says that, you know, regardless of how much money HHS, Health and Human Services, has, they can't support the use of these funds for the creation or destruction of embryos, regardless of whether or not the embryos would be used or destroyed. You know, if they're created by in vitro fertilization, the family no longer needs them because they've been able to get the number of kids that they want. They're not going to store these, in perpetuity. They're going to, at some point, get rid of them. Normally, they're going to be stored in, like liquid nitrogen. But when they're not used, the question is, what should be done with them? And this Dickey-Wicker Amendment says, we can't use them for the creation of stem cell lines. Now, a number of cases have tried to appeal this. And in the Shirley versus Sebelius appeal, Sebelius was Secretary of Health and Human Services. They basically said that this amendment doesn't prohibit research on established stem cell lines. And so we can't create new ones by creating or destroying human embryos. But it at least says that we can use some of these federal funds to continue stem cell research on lines that have already been created. And so it's a little bit of a slippery slope when we take a look at it. But it basically says we can't create new embryos for doing that. Now, if we take a look at the changes that have gone through, George W. Bush, president from 2001 to 2009, basically went with kind of that agreement and said, okay, we can allow federal funding, again, funding through the Department of Health and Human Services, for research on existing stem cell lines, stem cells that were created prior to 2001. And so this is before kind of those requirements went into place. So we can continue studying stem cells, but only on those 21 stem cell lines that were created before the rules went into effect. Barack Obama, president from 2009 to 2017, has expanded that a little bit to allow for the funding of stem cell lines created after 2001. And so this would be using stem cell lines that have been established, but not using federal funds to create the lines. So as long as the funds came from another source, or the stem cell lines were created without use of federal funds, we can continue to study them. In the 2016 21st Century Cures Act, it basically called for timely regulatory review of regenerative therapies using stem cells. And so as we begin to understand stem cell science a little bit better, how we can use it for the treatment of human disease, instead of having to go through a long regulatory kind of burden to get it approved, the 21st Century Cures Act ideally is going to allow for kind of a more rapid review in the hopes that we can get some of these treatments, these human stem cell treatments, out to the patients that need it relatively quickly. And again, these are the regulations that we have in place up to 2017. The Trump administration has greatly limited stem cell research. And so in 2019, the Trump administration ended fetal tissue research by government scientists and placed greater restrictions on academic researchers. So these are the researchers at colleges and universities that are seeking funds from the National Institute of Health. So again, federal funds administered through the Department of Health and Human Services. They now have restrictions on them for studies involving fetal tissues. They've also required the establishment of an advisory committee that has to go through and approve and review and approve all human embryonic stem cell research, even that's being done on existing cell lines. And so basically we had the Obama administration kind of opening it up, the Trump administration kind of locking it back down, at least up until this point. Now, if we take a look at this, the National Institutes of Health, again, as the primary provider of biomedical research funds, primary supporter of biomedical research within the United States, they basically, within their guidelines, are going to allow the use of funds on established embryonic stem cell lines, as long as these stem cell lines were created for reproductive purposes using non-federal funds. So again, looking at the Dickey-Wicker Amendment, what basically says we can't use federal funds for doing that. If the embryos or embryonic cell lines were created from embryos that were donated freely with proper informed consent, and so again, the parents are aware of everything that was involved, what was going to be done with these cells, and freely donated them for their use. But the National Institute of Health, again, within these rules that we have in place, will not pay for the creation of or research on embryonic stem cell lines from an embryo no longer needed that was created by in vitro fertilization. So again, in order to create these stem cell lines or to do research on the stem cell lines, they have to be these approved, previously established stem cell lines. So again, created before the restrictions went into place. So that's kind of National Institutes of Health, kind of taking a look at kind of pretty much research on the stem cell lines. The FDA, the Food and Drug Administration, is the agency within our federal government that's going to look at this research that in many cases has been supported by, biotech firms or pharmaceutical companies or the National Institute of Health through their grants to colleges and universities that are, professors studying these types of things. The FDA is going to look at that research and they're going to oversee what are referred to as the vaccines, the blood, and the biologics. And stem cell therapy falls into this category because they're going to be reviewed as a biologic. They're going to be seen as a tissue or a tissue product that needs to be approved prior to use within human patients. And so we've got, the cells that are being produced in stem cell therapy and what they're going to be doing to produce their products to treat the disease. All of this falls within the category of what the FDA is going to be overseeing. And so the National Institute of Health is going to be hopefully supporting research on the stem cells to be able to better understand them and to be able to understand how to use them in the treatment of disease. Then they're going to use that research to get approval by the Food and Drug Administration. Okay, so if we take a look at this, the Food and Drug Administration, as we said, regulates human cells, tissues, and cell and tissue-based products. And originally that was because of fears of contamination of these biological products. And so again, in the 1980s, there was a lot of fear about the virus associated with AIDS, HIV, being in the blood supply and potentially causing the transmission of disease through a blood transfusion. And that was, the case in the 80s where we didn't have good ways in which we could test for the blood. We now can test for HIV, but there are also, again, continued risks associated with other viral diseases like hepatitis, which we talked about previously within the course. And so the expectation is that the people that are doing the research on these stem cell lines and the people that are potentially using it for the treatment of human disease are going to be following good tissue practices to ensure that the cells remain free of viruses, that they're continually being monitored to make sure that they don't have infectious agents that could be transmitted to the patient if they were to receive stem cells or stem cell treatment with this. Within the FDA, it's regulated within the Center for Biological Evaluation and Research. And so we have a mechanism in place that kind of evaluates whether or not this is going to be approved for the use of stem cells in human subjects. And so what we can see is that pretty much across the United States, in 2016, we had stem cell treatment clinics, except for North and South Dakota, kind of over here, kind of in the north central region within the United States. Now, some human cells, tissues, and cellular or tissue related products are exempt from the FDA regulations as long as they're able to show that they've used those good tissue practices. And so they've gone through and able to demonstrate that they haven't got stem cells that are infected and they're using good practices, so they're not going to become infected, they're not going to cause disease in the patients. And so what we can see, again, stem cells, not embryonic stem cells, which we were talking about previously with the regulations, but stem cells can be used and it'd be exempt from FDA regulation if they're minimally manipulated. And so you're not going to be changing them in some way. So you're not, changing their genetics or providing chemicals with them or doing something else to manipulate them in some way. If they're not combined with another agent or samples from another source. And so you can't, take your stem cells that have been isolated from your body and combine them with embryonic stem cells, or combine them with stem cells from another patient, or from a pig, or something like that. It's got to stay human stem cells, so we don't have the risk of zoonotic diseases, or ideally the risk of disease from another donor. And if it's for autologous use. And so autologous means that we're going to be harvesting cells from a patient and then putting them back in without manipulating them, without combining them into the same patient. And again, that's similar to what we looked at, I think 2 days ago in the lecture, where we talked about the treatment of like joint diseases in dogs and horses, where they isolate the fat cells, pull out stem cells from the fat, and then inject it back into the patient, injecting their own cells, minimizing the risk that we're going to have tissue rejection, that immune response that we keep talking about it. The FDA has loosened this up a little bit to say that we can do that with a first or second degree relative. Basically, your parents or your siblings or your offspring would be able to donate cells as long as they match, have a good match for you. We don't have to worry about getting FDA approval. There is an approval that, or not approval, there is an exemption, so you can use it for reproductive uses. And so I'm not quite sure how this would work, but we can, collect human cells and do, this would be the equivalent of in vitro fertilization, which is already approved. Now, as we said, the good tissue practices are going to be important, because we want to make sure that if we take these cells and put them into patients, we're not going to cause those patients to become even sicker. We're not going to cause them to catch diseases. And so because of that, we're making sure that the donors, if we're getting it from one a family member and putting it in, we're not going to have these transmittable diseases. So we're going to screen it, we're going to test the cells, and we're going to make sure that they don't become contaminated during the process of preparing them for the injection. Now, this is related to the good manufacturing practices. That was good tissue practices up here when we're working specifically with cells and tissues. Good manufacturing practices are basically regulations that the FDA, the Food and Drug Administration, has in place to make sure that if we're a company producing drugs, producing either name brand drugs or generic drugs, that we're going to be doing it in a way that we can ensure the identity, the strength, the quality, and the purity of the drug product that we're going through, making sure we're using the right raw materials that aren't contaminated, that we're using our equipment, it's calibrated, so we're measuring around the amount that we need, and we're mixing it properly, we're using good procedures, and we're continually testing our samples to make sure that what we think we have and what we're going to be using on these patients is what we truly have. And again, this is something that we want to make sure when we're producing, again, brand name drugs or generic drugs. If we take a look at this, again, within the Public Health Safety Act, getting into a little bit more of the regulations associated with it, this regulates the licensing of biological products. Another example of a biologic product would be the serum treatments that people are talking about, taking patients that have been exposed to COVID-19, have recovered from it, isolating the antibodies, kind of the immune system proteins within the body of a patient that's recovered, and using that serum to treat patients that are suffering from the disease. And so as it relates to stem cell technology and stem cell treatment, we're going to be going through and making sure that if we're going to be doing something that's highly processed, altering it in some way, used for other than their normal function or combined with other things, we now have to get approval by the Food and Drug Administration. And normally what this means is we have to do that investigational new drug application. And so if we take the cells out and put them into the same patient, the patient is both the recipient and the donor, we don't have to get approval. If we take the cells out and maybe purify them a little bit and put them back in, we don't need to get FDA approval. But if we're going to go through and alter them in some way, change what it is that they're going to be doing, that now requires that we get FDA approval, and we have to go through that new drug application process to be able to show that what we're doing is based on good science, and there's a reason for us doing that. Now, an example of this is REGENX. REGENX is a US-based company that uses the patient's own stem cells for treatment. So again, as we've looked from these guidelines, as we've talked about it, this means that REGENX within the United States is approved to do same-day procedures. So normally what they're going to be doing is they're going to take a bone marrow biopsy, a little bit of blood from the patient, process the cells minimally, so maybe isolate the cells a little bit, purify the cells a little bit, but not change what it is that they're going to be doing. And then we can inject these cells into a region of the body needing repair. Normally, similar to what we saw with dogs and horses, primarily this is injecting them into joints or tendons and ligaments and bones to repair kind of a region in that way. Again, similar to what we see with veterinary medicine. Now, the FDA is taking a look at this idea that your cells equal drugs policy is going to prohibit the culturing of the patient's stem cells prior to implantation. And so the REGENX position is that they're your cells. The FDA position is that they regulate chemical drugs, and if we're going to alter our cells, even though our cells, if we're altering them in some way or altering what the cells are producing, that is going to be subject to regulation. So if we're altering them, if we're changing them, if we're culturing them for a period of time, not doing that same day, injecting them back in, this now becomes a drug. And if it's a drug, then it has to be approved by the FDA. And in order to do this, it has to go through that investigational new drug application process, show the evidence, be able to enter it into a clinical study, clinical trial, and basically means that a lot more study needs to go into these things before it can be used readily within patients. Now, having said that, we've got these regulations then within the FDA and these rules on what we can do with stem cells. We often see areas of stem cell medical tourism. I talked about the football player Peyton Manning, who went, I think, to Sweden about 10, 15 years ago to get stem cell therapy because it wasn't available within the United States. Regenex, that company, 2 slides ago, Regenex, that US company using the patient's own stem cells for same-day procedures, has an offshore branch. Regenex-C is located outside of the United States through independently owned and operated medical service providers that are in locations that are allowing for these cultured cells, so autologous still, taking the cells from the patient, growing them for a period of time, and then putting them back in. Not approved within the United States, but approved elsewhere. And so this is a separate kind of related company to Regenex, but they basically are doing this, outside of the United States, and therefore patients may decide that this is what they want. They're going to have to go outside of the United States in order to be able to get this treatment. And again, these procedures are not approved by the US FDA for use within the United States. So that's why we're looking at medical tourism, because patients have to leave the country to be able to get these treatments. And again, they're going to come back into the US and potentially have to be monitored if there are problems that need to be occurring. And that, again, is a big fear, that if we inject these cells in, what is the long-term impact? What is going to happen? Are they going to form tumors? Are they going to be lost and the patient's going to have the disease come back? All of those things are being studied right now. And so hopefully this gives you kind of a good overview about some of the rules and regulations associated with both the insurance coverage for stem cell technology, as well as experimental medical technology, as well as some of the rules and regulations for the study of stem cells and the use of stem cells for treatment. What I've got for you for readings for today, again, are a little bit more information about FDA regulations on stem cell-based therapies, health insurance coverage for clinical trials, and another article associated with insurance coverage and the cost of clinical trials. Again, you want to be informed if you're going to be involved with a clinical study. And that finishes up the lectures for today. Come back for part C, part 3 of these lectures, where we're going to talk about the activities for today. Thank you and have a great day.